So during nutrient excess, you're going to make triglycerides, or fat. So that's indicated by elevated glucose levels, which stimulates the pancreas to release insulin into the blood. Now insulin will react with the receptor on its target cells and induce expression of triglyceride synthesis genes. However, recently it's been discovered that another molecule called carbohydrate response element binding protein, or CHREBP, was discovered to do this also. And it's activated by glucose. So if you have glucose in the blood, it's going to be transported into the cells and broken down. The metabolites of glucose then activates CHREBP, and it will translocate into the nucleus and heterodimerize with its binding partner, MLX. Now this complex will then bind to DNA and induce transcription of these lipogenic genes to create fat. CHREP is expressed in a bunch of different tissues, such as skeletal muscle and fat. We know that it has a really important role in the liver in glucose and fat metabolism. However, discovering what is functioning in these other tissues is hard in animals because of a lack of genetic tools. Drosophila is a great model to use because a lot of the metabolic pathways are highly conserved in this animal. It also has lots of genetic tools that we can use to study it. In Drosophila, the homologs of CHREBP is Mio, and the homolog of MLX is Big Max. So like mammals, there's lots of metabolic tissues in Drosophila as well. So our lab looks at what Mio's function is in these tissues. And so previously, our lab has looked at Mio in the fat body, which is a fat-like tissue, outlined here in red. So Sasu et al. Discovered that Mio promotes triglyceride storage in the fat body, and interestingly, it also promotes feeding. So we all know that the brain is also involved in the regulatory process of feeding. So that's what I'm looking at.